Welcome to the deep dive. Today, we are strapping in to examine one of the most stubborn yet least understood challenges in modern oncology, and that's the environment that cancer builds around itself.
It's such a critical area. So often treating cancer fails, not because we can't kill the cancer cells themselves, but because we can't get through or, neutralize the ecosystem they create. There's this powerful analogy that cancer is like a wound that never heals.
Exactly. And if the tumor is that unhealing wound, then the major architects of that whole environment are the stromal cells, specifically fibroblasts. And when they get tangled up with cancer, we call them cancer-associated fibroblasts, or CCAFs.
Right, and CCAFs are, at their core, just these essential mesenchymal cells. There's the structural workers that, hold our tissues together. But when a tumor shows up, they get corrupted, they go rogue, and they become this key part of the tumor's defense, its growth mechanism. For a long time, the big puzzle has just been their sheer diversity, their heterogeneity.
Early attempts to categorize them sort of give us three main buckets, right, based on what they do. You had inflammatory CCAFs, ICAFs, the myofibroblastic CCAFs, MICAFs, which are the main guys producing that stiff protective matrix. And then you have this odd one out, the antigen-presenting CCAFs, or apCAFs.
And the apCAFs are, they're truly fascinating, almost paradoxical. They're supposed to be structural cells, but they express these immune-related molecules. Specifically, they express major histocompatibility complex class 2, or HLA-2 in humans. This is a job traditionally reserved for immune cells, and lets them present antigens and regulate T cells. So giving a structural cell an immune function like that is revolutionary, but honestly, until very recently, their true origin and role were, they were murky.
That murkiness is exactly what our source material, this massive new study, set out to solve. And to do this, researchers didn't just look at a few tumors. No, they integrated data on an unprecedented scale. We're talking 532 human samples, encompassing over 2.5 million cells, just to create this comprehensive fibroblast molecular atlas across 15 different tissue types.
The scale is the story here. And because of that scale, they didn't just find one kind of apCAF. They revealed a startling discovery that really it completely changes the blueprint of the tumor ecosystem.
Okay, so let's unpack this. Our mission today is to reveal that apCAFs are not this single population, but are actually two distinct geographically specialized populations, and that these two populations are critical drivers of tumor progression and therapeutic resistance through, get this, the deployment of a single powerful protein. So jumping right into this monumental atlas, the very first thing the researchers noticed was that this previously uniform apCAF population, it split cleanly into two robust, molecularly distinct clusters. And they were present in almost every single cancer type they looked at.
Exactly. Which immediately tells you these aren't some weird anomalies. These are fundamental parts of the system. And when they dug into the gene signatures of these two groups, they found fundamentally different cellular identities, different origins. They're now defined as M-apCAFs and F-apCAFs.
Let's start with the M-apCAFs. Where did these specific structural cells come from?
M-apCAFs are the mesothelial-like apCAFs. The mesothelium, that's the lining of our internal body cavities, like the peritoneum, which is where abdominal cancers so often metastasize. And these cells express characteristic mesothelial genes like MSLN and KRT19. This profile confirms they are, local residents that have undergone a transformation.
So they're like the neighborhood residents who suddenly decide to turn their homes into fortresses for the invaders. This is what researchers call a transition, isn't it?
Yes, exactly. A mesothelial to fibroblast transition. Think of it like a local skin or lining cell that, when it's irritated by the tumor, it just completely changes its job description. It stops being a passive lining and turns into this active building block for the tumor's defenses. And interestingly, M-apCAFs also express CD24, which is a gene linked to innate immune regulation.
Okay, so the M-apCAFs are the local recruits. What about the F-apCAFs?
The F-apCAFs are the fibrocyte-like apCAFs, and their origin is completely different. They align with features of fibrocytes, which are migratory cells. That means they're derived from the bone marrow, making their origin hematopoietic.
Wait, so they're not local residents at all. They're travelers arriving from the circulation.
Correctly. There are these molecular hybrids. They express a classic white blood cell marker, PTPRC, that's the gene for CT45. but they also express standard structural markers, like the collagen gene COL1A2. So they're immune cells kind of trying to play the role of structural fibroblasts.
Right.
And we know fibrocytes travel to sites of inflammation or wound healing.
That makes so much sense. So M-apCAFs are the local builders, and F-apCAFs are the specialized migrating emergency crew. Now, how do we actually confirm these cells' life stories? How do they know this for sure?
So researchers use something called pseudotime analysis. It's a really sophisticated computational method. Instead of watching cells age in real time, which you can't really do, they take a snapshot of many cells in different stages of maturation, and then they use these algorithms to sort of reverse engineer their developmental path, basically figuring out the life story of these cells from start to finish. And this confirmed the duality. Both M-apCAFs and F-apCAFs mature along their own distinct paths, and as they do, they both turn up the volume on those HLA genes.
Which confirms their antigen-presenting function. But here's the absolutely crucial part, the mechanism that explains why these cells are the enemy. If they're presenting antigens, they're essentially showing the immune system the target. You'd expect T cells to activate and attack, but they don't. Why?
Because the apCAFs are saboteurs. They are antigen-presenting cells that intentionally lack a necessary second handshake, the co-stimulatory signals. They are deficient in key molecules like CD40, CD80, and CD86.
Okay, give us an analogy for that second handshake.
Okay, imagine the apCAF is showing a T cell a wadded poster of the tumor antigen. That's signal 1. But to actually launch the attack, the T cell needs the second signal. It needs the go button, the keys to the car, the weapon. If they show the wanted poster but don't give that go signal, the T cell doesn't activate. It freezes, or even worse, it enters a state of tolerance. So the hypothesis is that this deficiency leads to the induction of regulatory T cells, which are massively immunosuppressive. They stop the immune response before it can even start.
So we've got two different populations, one local, one migratory, both able to present antigens, but both designed to hit the immune system's brakes. This immediately makes you ask the next question. Do these different origins mean they have different jobs inside the tumor?
To answer that, the study used this cutting-edge methodology, high-resolution spatial imaging. So instead of just grinding up the tumor and sequencing cells, they were able to physically look inside the tissue and map the location and gene expression of individual cells, seeing exactly who they were sitting next to.
That's the key, microanatomy. So where do the F-apCAFs, the travelers, where do they choose to settle down?
F-apCAFs are the immune gatekeepers. They consistently settle in lymphocyte-enriched niches. You can think of these niches as temporary immune hubs or even rudimentary lymph nodes inside the tumor environment. They're often called tertiary lymphoid structures. And in cases of peritoneal metastasis, or PM especially within the ICMS3 subtype of colon cancer, which is known to attract a lot of T cells, F-apCAFs were the most abundant cell type in these immune hubs.
Wow, so they're sitting right in the war room. What are they broadcasting from that strategic position?
Well, their messaging is deeply complex. It's a real double-edged sword, which reflects their unique immune fibroblast hybrid nature. On one hand, they secrete CCL5, which is a strong chemoattractant that draws more T cells and monocytes into the area. They're actively recruiting the immune system. But at the same time, they're secreting immunosuppressive genes like LAR1 and critically this protein SPP1. They basically lure the immune cells in, then immediately start trying to regulate them or even suppress them.
It's an evolutionary trap. Okay, so let's turn to the M-apCAFs, the local residents that change their identity. What is their job and where do they build their fortress?
The M-apCAFs are the tumor protectors. Their job is to shield the cancer. They are located proximal to the cancer cells themselves, and they often sit right next to the remaining mesothelium. This physical location strongly supports the idea that they arise from that local mesothelial to fibroblast transition right at the tumor edge.
And what does that proximity do to any immune cells that try to get close to the tumor?
It just exhausts them. When the researchers analyzed the T cells found in these M-apCAF niches, they were highly exhausted. They expressed a whole battery of suppressive and exhaustion markers, things like TIGIT, CTLA4, LA3, PDCD1, which is PD1, and FOXP3. This confirms M-apCAFs are driving this acute, localized immunosuppression right at the tumor interface. They're forming an active shield.
This spatial map is just so powerful. Let's pause for a quick recap of this division of labor.
Absolutely. The research really gave us the definitive split. First, two faces. We have mesothelial-like M-apCAFs, the local builders, and fibrocyte-like F-apCAFs, the migratory travelers. Second, the F-apCAF role. They cluster with lymphocytes in the immune hub, where they recruit cells but then try to regulate them from the periphery. And third, The M-apCAF role. They cluster directly next to the cancer cells, acting as the ultimate immune suppressor and physical barrier at the tumor edge.
So since these two populations are so strategically positioned to manage the entire microenvironment, they must be coordinating their defenses. What's the single powerful molecule both populations are using to drive such coordinated effects on the immune system, metastasis, and the tumor itself?
And this is where the whole story converges. It points directly to one molecule, secreted phosphoprotein 1, or SPP1. You'll often hear it referred to as osteopontin.
We hear that name SPP1 a lot, and it's almost always associated with bad outcomes in cancer. What makes it so destructive in the context of these apCAFs?
SPP1 is an acute phase protein. It shows up during inflammation, during tissue remodeling. It's a very, very strong negative prognostic marker in cancer because it fuels fibrosis and metastasis. And what this study showed is that apCAFs are the major suppliers. They exhibit substantially higher expression of SPP1 compared to every single other CCAF cluster they identified in the entire atlas. And its production just increases as both M-apCAFs and F-apCAFs mature.
So high SPP1 production is the signature of an aggressive apCAF, whether it's a local builder or a migratory immune regulator. What are the consequences of all this SPP1 secretion?
SPP1 is a genuine multi-tool weapon for the tumor. The evidence shows it facilitates primary tumor formation. It significantly promotes metastasis, especially peritoneal metastasis, or PM. It drives desmoplasia. That's the dense fibrotic scarring that blocks treatment. And critically, it increases therapeutic resistance.
Let's look at that evidence, starting with peritoneal metastasis. How did they prove that apCAF-derived SPP1 was the primary engine of metastasis?
They used a really powerful experimental approach. First, they looked at mice with colon cancer-derived PM. And when they performed a knockout of the host SPP1 gene, meaning the stromal and immune cells in the mice couldn't produce the protein, the result was a substantial, measurable reduction in both PM development and ascites, that fluid buildup. And they confirmed this by simply treating the PM-bearing mice with an anti-SPP1 antibody. Same effect.
Okay, that's compelling on the systemic level, but how did they prove it was specifically the apCAFs causing the harm?
They isolated the apCAFs. And they showed that the SPP1 secreted by just these isolated cells significantly increased the colony-forming and invasion capabilities of the cancer cells in the dish. And that effect was completely blocked when they added the anti-SPP1 antibodies. It definitively isolates the apCAF as the critical source of that metastatic signal, using SPP1 as the chemical handshake.
Wow. That brings us to pancreatic ductal adenocarcinoma, PDAC. This cancer is notoriously resistant to chemotherapy because of that dense physical tumor armor, the desmoplastic barrier. So how do the M-apCAFs and SPP1 contribute here?
In PDFs, the spatial analysis showed M-apCAFs were enriched in the SPP1 positive areas, confirming they are creating that protective niche right next to the tumor. The study then showed that SPP1 derived from the apCAFs actively promotes the differentiation of other structural cells, these pancreatic stellate cells, into MICAFs.
So the M-apCAFs are using SPP1 as a signal to hire more vectors, creating a vicious cycle of fibrosis and barrier formation.
Precisely. The SPP1 is the instruction manual that tells the tissue to build the armor. And this has huge implications for treatment failure. Targeting host SPP1 in mouse PDA models didn't just reduce tumor growth, it's significantly increased T-cell infiltration, and perhaps most clinically relevant, SPP1 blockade successfully reversed chemoresistance to FOLFIRINOX. That's the intensive chemo for PDAs and cancer cells when they were co-cultured with apCAFs.
Wow. So by targeting this one molecule, SPP1, you were tackling three major hurdles: tumor growth, the physical desmoplastic armor, and chemotherapy resistance. The molecule connects the microanatomy right to the clinical outcome.
This deep dive has really provided an unprecedented resolution. We now have this clean framework. apCAFs are two populations with tailored yet coordinated functions.
Right. The F-apCAFs, the travelers, are the complex immune modulators. You find them in the T-cell-rich areas. They recruit, but they also regulate and suppress, using SPP1 as part of their arsenal.
And the M-apCAFs, the local residents, they're the immediate tumor collaborators. They build the SPP1-rich niches right next to the tumor, driving fibrosis and chemical resistance. They make the cancer physically impenetrable and chemically immune to treatment.
And this research also helps us resolve some of those prior conflicting reports in the field, doesn't it? Earlier studies often struggle to fully characterize apCAFs.
That's exactly right. If previous researchers were focusing their studies on, say, tertiary lymphoid structures, they were likely only capturing the F-apCAF activity, which expresses markers like C1q. They were completely missing the M-apCAF lineage, the local SPP1-driven tumor protectors that are actually creating the immediate barrier to treatment. We now have a complete picture of the attack strategy.
So let's talk therapeutic relevance. Identifying SPP1 as the common link is a huge deal. Why should this excite a patient or clinician right now?
Because targeting SPP1 is a genuine, multi-pronged approach. First, you degrade the physical armor, the desmoplastic barrier, making it easier for drugs and immune cells to get in. Second, you are removing the key signal that's driving chemoresistance. And this is particularly relevant for these really tough cancers like PM and PDA.
And what makes this fast-track material?
The clinical momentum is already there. Humanized SPP1 antibodies are already in clinical trials for other inflammatory and fibrotic diseases. This means that the development pipeline is already significantly advanced. If these results translate to the clinic, we could potentially fast-track SPP1 blockade as a crucial synergistic therapy, a way to sensitize these notoriously resistant tumors to existing treatments, targeting one stromal molecule to clear the path for conventional treatments to finally work.
This detailed spatial map is such a powerful reminder that microanatomy truly dictates macro treatment success. We can't just target the cancer cells, we have to disarm the architects of the environment.
And that leads to a final provocative thought for you, the listener. We've established that M-apCAFs are the tumor protectors and F-apCAFs are the immune gatekeepers. Now that we can tell them apart, should future therapeutic approaches focus on, say, the total elimination of the M-apCAFs, the most harmful resistance-promoting lineage? Or should we aim for a more nuanced approach to maybe reprogram the F-apCAFs, turning them from suppressive gatekeepers into fully activated, immune-stimulating antigen-presenting cells to boost the body's own defense system?
The choice between elimination and sophisticated reprogramming, a profound challenge offered by the two-faced fibroblast. Thank you for joining us for this deep dive into the immense complexity and strategic vulnerabilities hidden within the cancer microenvironment.